Welcome to Ferment Radio. I am your host Aga Pokrywka and my guest today is Mindaugas Gapševičius. Miga is an artist, facilitator and curator based in Berlin, Germany and Vilnius, Lithuania. He is currently conducting a PhD research at the Bauhaus University in Weimar, where he holds an artistic associate chair. Together with colleagues from the TOP Association, he stands behind TopLab, the first community-based biolaboratory in Berlin. He also co-established AltLab, an open research laboratory in Vilnius. I was thinking actually that I think that we've met for the first time in 2008 when I was an exchange student in Vilnius. And I remember it was still a time when, new media art was super big and talking about science and art a bit lesser, maybe. And I perceived you as a person who worked with technology and art. And now in your practice, this element of living things, of biology is much stronger. And I actually never asked you, how did it happen?
Yeah, interesting that you reminded me 2008, whatever. So I guess it was a workshop with John Hopkins, perhaps?
Exactly.
Yeah, interesting. So, at that time I was still fully into networking and computer technologies, and programming for me was kind of my practice. And somehow at some point I got a feeling like, okay, so I can't be on the level of artificial intelligence without being present in life. So for me, kind of the transition happened pretty naturally. So I developed computer stuff, programming, and then I started to include at some point physical reality, and it went into bio stuff. So now I can kind of combine computation and biotechnologies all together. I think the cycle became complete in a way. So I can think of artificial intelligence in terms of, or from the perspective of a human being, which makes much more sense as to think of artificial intelligence from the perspective of computers.
Mm-hmm. I just realized that maybe I should have asked you at the beginning a question like how you would describe yourself and what you do and who you are.
I introduce myself usually as being artist, and I do doctoral research at Bauhaus University of Weimar, where I also teach, and I also work quite a bit with projects, let's say, which involve people, organisms, and tools. So I kind of try to think from a philosophical perspective and practical perspective how things interact with each other. Things I have in my mind now, like also living organisms, including microorganisms and other organisms ourselves.
Can you tell more about some of your maybe recent or maybe the most important for you works involving microbes?
Yeah, I have started with Introduction to Posthuman Aesthetics, probably project back in 2016, I think, and I started to develop kind of toolkits for the work with microorganisms, for DNA, for electricity, kind of diverse toolkits. And one of those was a toolkit for work with microorganisms. It would mean isolating microorganisms, growing into the colonies, and seeing how they behave, interact between different colonies. And then probably the next biggest bigger project regarding the work with microorganisms was and still is a proposal for microbial therapy. And this project is involving the work with microorganisms which would be functioning as probiotics. It is about fermented food with precise organisms you want to be consuming afterwards. So right now I'm kind of developing this project and working with diverse microorganisms, diverse people, and seeing how actually our perception changes while consuming very much specific microorganism.
And what kind of microbial therapies you were thinking of so far?
Well, the main organism we have been working is Streptococcus and Lactobacillus, different strains, let's say. And right now we work also with Leuconostoc and diverse yeasts, but we are not yet far enough with those. Thinking of Lactobacillus and Streptococcus. So these guys thrive in yogurt. So the idea is to isolate microorganisms from yogurt or from kimchi or sauerkraut and grow it into the fermented foods, let's say, with a focus on this precise organism, and then consume for a couple of weeks and see how the perception changes.
So the role of a toolkit is to give instructions how to recreate the process you are documenting in the toolkit. Why do you feel this kind of approach is important? Because you could easily just, I don't know, create those works without actually giving any instructions how to redo similar process.
On one hand, if you would talk to someone who doesn't have any idea how to isolate microorganisms and how to grow them into the colonies or into fermented foods, people would think that, oh, it's too complex, too complicated. But as soon as you have a toolkit, it becomes very simple. So you have a couple of tools, you have a sample of microorganism. It makes the life much more easier. And you include all instructions and you include all the tools with what you could continue working as a scientist or as a artist or as a researcher, whatever you are working on. So the things become much more simplified, and you get the idea very quickly what is the toolkit about, what is the bacteria about, what are the related resources about. So this is one part, perhaps educational. And the other part would be artistic. So the toolkit is an art object itself. So you can exhibit it in the exhibition space, for example, or you can give to someone to have it at home. And the idea on top of that is that the audience, in this case, audience either at the exhibition space or at home, is interacting with tools and with organisms. So the audience becomes part of the artistic object and adds on top of the artistic object another layer, another value onto it. So it is a kind of a setting for collaborative artistic work.
To me, it also sounds like an approach to demystify science or things which actually are represented in a society as something more professional or something you need special devices for. This conclusion or this observation is maybe also impacted by the fact that you are not just creating toolkits in your art practice, but you are also standing behind at least two, right? Open labs for like a bio art experiments. Is it so? Do you feel the connection between those two?
Yes, of course. Yeah, it is a very precise connection. On one hand, toolkits are for the work, for the individual work, let's say, more. And if you keep on running biolabs or labs, let's say, so it is including a little bit more space, a little bit more tools, and you can work together with people. So it becomes, instead of one to toolkit, it becomes many to tools. So it is a little bit broader perspective, but I think laboratory is exactly the same as a toolkit. It's just the different scale. And regarding your first question or comment regarding demystification, it's it is in there, and it is a very important part. Of course, in the artistic practice or my artistic practice, I think we have too much information regarding theory and philosophy, and if we would think of pandemic, COVID virus or so it's a lot of information for which for us seems to be ungraspable, too far away. But as soon as you start working with those things, let's say having a toolkit next to you, you start understanding this world from a practical part. So it's a kind of different perspective. It's not theoretical, but it is practical perspective. And I think this demystification happens when you start actually dealing with things practically, not only thinking about them.
We've mentioned here before the spaces or labs you are co-creating, or you've been involved in creation of them. And I meant here, from this what I know, the top lab and alt lab, right?
So Alt Lab and Top Lab. So these are my initiatives. I was initiating these labs in both Berlin and Vilnius, and they are kind of places for interdisciplinary research. So people who are working in these labs are not necessarily artists, but we have diverse people, scientists, artists, theoreticians, people who work with technologies, students, different people in a way. So in the end, it becomes a collaborative space for doing diverse things.
Do you have any like a specific equipment in a top lab in Berlin?
I think we have this basic equipment which is needed for sterile work, let's say, or which is needed for work with molecular biology. So the tools would include laminar flow hood, thermocycler, then tools for electrophoresis, those which are not available at home.
Why people should do those things? Why we, for example, want to encourage somebody to, I don't know, create whatever, cellulose from microbes? What's the point?
I think it involves many different points in here. The collaboration in itself, if you would think of artistic practices, so we could think of collaboration or collaborative moments from the perspective of fluxes, for example, or relational aesthetics. So kind of performative activity which involves audiences into the art production, let's say. I don't like the word production, but it is not only the artist anymore, as it was used to be in visual arts, but also the audiences. And in this case, the space is being inhabited by more people than one. So in this case, museum context becomes less important, but more important becomes physical space or social space, which creates on top of that new ideas and new developments. I think this is the most important part in collaboration. On the other hand, collaboration is needed because of the complexity of our lives. So, you being an artist, you wouldn't be able to think in terms of contemporary art without thinking in terms of diversity of tools available. So, think of computers, think of sciences. You alone, you wouldn't be able to grasp all the things all around. And the third point in here is probably being able to think of feedback loops and the influence to your thinking from the environment, kind of Uxkulian Umwelt idea, and be able to perceive this influence from environment and be able to give it something back. So we talk also about feedback loops between things, between humans, between organisms, and between tools. Yeah, so therefore I think the collaboration is much more complex issue, and it is needed in order to be able to develop something on top. You can't ignore it because you are a social being and you live together with other let's say, organisms.
Yeah, and it seems to extend like beyond human species because in, I don't know, the labs, it's not just collaboration between humans, but also bacteria or other species. And I think that's probably the most kind of, yeah, this is beyond Fluxus, I guess.
Yeah, it's true. So Fluxus didn't go that far away, of course. What they have added on top of imaginary art was experimental art, and they were already kind of using diverse experimental forms, so like cooking sessions, or interactive playing installations. But what we have beyond of Fluxus, we are able, I think, to contemplate from the perspective of the other. So we could think of, let's say, interaction between diverse species, including microorganisms and ourselves. And on top of that, I think Fluxus was still kind of egocentristic a little bit. So the artist itself would be the artist. And what we have right now is more a collaborator in the bigger setting. And to say the truth, I really like this word you mentioned, collaboration, between different species. And of course, this term is not being used for the first time, and you have pointed to it, but it is very interesting that these terms come also from philosophers. And right now, as I mentioned probably already, that I work on a book, Share the Habitats, so the set of exhibitions I have been working on. And one of contributors to the book is Andrew Pickering, who also kind of tried to describe this kind of change between what is happening nowadays in the exhibition, let's say, framework. So instead of having collaboration between people, like imagine theater play or something, we have now in our installations also organisms which become part of experience, part of the artistic installation.
How actually we can interact with microorganisms, like how we can experience them?
It's a good question, of course, and many people, including my professor Ursula Damm and scientists, would criticize me because of this collaboration between microorganisms and myself, let's say, or humans. But I don't agree with them on this point, because if thinking from the perspective of probiotics, so all industry, medical industry, which is developing probiotics, do not develop them just because of developing and because of business. It should make some impact for our organism. And also, there are many scientific papers which deal about the impact of microorganisms to ourselves. So that would include, of course, the microbiome, microbes living within our guts. So thinking further, if you as a consumer would consume probiotics of one sort of bacteria or yeast, you would be able to experience some change after a certain time. So let's say after 2 weeks. Some people might be continue thinking that this is imaginary, this is social construct, but if you would try to experience your feelings yourself, being alone, you will notice that something is changing with your body, and is not only a speculative idea, at least from my perspective. So thinking of changing our microbiome while consuming precise microorganisms, I would say this is a kind of collaboration which comes to my mind. So very direct collaboration. So bacteria is changing our cells, our perception, and we change also bacteria in a way that it starts mutating depending on the umwelt.
You mentioned about that your professor or some other people might not be understanding this in the same way as you do. So what's their stance, or what is this thing that they disagree in your approach with?
Well, bacteria, if I would think now of Ursula Damm critique, so bacteria probably would be for her too small in order to be able to collaborate. And from the perspective of my collaborator, Auksė Gaižauskaitė, who helps me with scientific things, she would think, well, we need to prove it, scientifically prove it. From my perspective, I can critique their approach, which is nothing wrong, which is developing further into further collaborations and development of further ideas.
Coming back to art practice, and everybody can be an artist, therefore blurring this border between artists and other people. I think in this case it's like blurring the border between human and non-human. At the end, these divisions are just like a conceptual framework, nothing more than that.
Yes, exactly. These are just conceptual frameworks in order for people to understand where they live, which is pretty much scientific understanding or approach. But as you started to mention about conceptual frameworks and constructs, I started to think about Deleuze and Guattari idea of state in between, or the rhizome, which is which is always in between. So what is human? How would you define human? So this organism which is Homo sapiens, or which is slightly different, let's say genetically modified human being, right? So what is human in this case? So these kind of questions, I think, exist in the society just in order to be able to call the things, in order to make the things to understand easier. But yeah, beside humans, there are so many species around the world which have also states in between, like protozoa, which are neither bacteria nor fishes, So something in between. So many species have so broad genome that at some point they are not any longer one species, but they have features of the other species.
But we can go even in this deconstruction farther, because if human, it's something which actually is not so easily to define, or there are many, question marks here, then also what is a creativity when we reflect on our bacteria or microorganisms? I think then we end up with, questions. So what does it mean to be creative?
Yeah, exactly. So creativity is, in general, I think, is the term which is used everywhere and all the time, and it makes no sense without defining creativity. But I think in my case, or if I would start thinking of creativity, first of all, I would say it's a kind of developing new ideas and implementing new ideas somehow. Uh, uh, would that be kind of artistic object, or would that be a technological object? It makes no much difference, but I would see creativity more as kind of reaction to what you have all around. So you are not satisfied any longer with this kind of table which stands in front of you. So you come up with a new form of the table so it would start making sense for you. So here I would see some kind of creativity.
In such a sense, microorganisms are super creative then, because they are like highly adaptable and extremely fast, actually reacting on changing like circumstances and environmental conditions.
Exactly. So yeah, I think that all organisms from this perspective are super creative because they are able to adapt to changing environment.
Wow. And what's waiting for you in the future, or what you are looking forward to regarding your work?
Yeah, it's always very interesting to dream of future. I don't know, but yeah, I have so many ideas I want to kind of implement, and I want to continue working with people. So, but yeah, in the end, it's about being able to kind of interact with your environment, including humans and non-human organisms, and also being able to contribute something to society and being able to take something back from the society.
Actually, what I found interesting about this podcast, that there is this time slot when you have the conversation with a person, but probably if you would have this if we would have a conversation yesterday, probably we would talk about something a little bit different. Probably if we would have this conversation any other time, it would be about a bit something else.
Of course, yeah, I don't have any doubts in here. So yeah, it is pretty much a momental thing. It depends on the impact of the environment, which has so many layers to think of.
Maybe it even depends on state of microorganisms in our
Of
stomach.
course, yeah, it is pretty much truth. I don't know now. I wouldn't be able to quote anything, but our thinking is pretty much dependent on microbiome, how healthy it is, for example, and how diverse it is.